Hi, my name is Kirsten Kennedy. Today is March 21st, 2021, and today I will be talking about the determination of gender, genes versus hormones for my week 13 portfolio. So chromosomes, each individual contains 46 chromosomes. They occur in pairs, or also known as diploid numbers. 22 matched pairs of autosomal chromosomes and one pair of sex chromosomes, which include an X and a Y, or an X and an X. The amount of DNA in a diploid cell is written as 2N, and when the egg and the sperm unite is when the resulting zygote contains a unique set of 46 chromosomes. These chromosomes directly develop into human body forms such as hair color and blood type. Sex chromosomes determine genetic sex. Genetic females have XX chromosomes. They inherit both X's from both parents. Genetic males have XY chromosomes, and they inherit an X from their mother and then a Y from their father. Presence of a Y chromosome determines if they are male. So even if it's an XXY chromosome, it will still be male. The absence of a Y chromosome determines if they will be female. In females, if there is an absence of an X chromosome, then it's considered Turner syndrome. And when an X chromosome in each cell of a female's body is inactivated and condensed into nuclear matter, that is considered a Barr body. The sexual differentiation occurs in early development. Reproductive structures do not begin to differentiate until the 7th week of development, and this is considered bipotential. They cannot be identified as male or female reproductively because earlier than the 7th week, you just cannot tell. Bipotential internal hematuria includes the Wolfram ducts, which are derived from the embryonic kidney, and the Müllerian ducts. One pair of ducts develop while the other degenerates as development reproductively proceeds. The bipotential external genitalia includes genital tubercle, urethral folds, urethral grooves, and avioscrotal swellings, and these develop into male and female reproductive organs as development goes on. The SRY gene. So the sex determination depends on the presence or the absence of the sex-determining region of the Y chromosome, or the SRY gene. So if there is a Y gene, then it determines that they are a male, but if there is no Y gene, then they are a female. So the presence of this, the bipotential gonads, develop into testes, and then the absence of this, under the direction of multiple female-specific genes, the gonads then develop into ovaries. So, male embryonic development. The SRY gene produces a protein that binds to DNA and activates additional genes. The protein product of these genes direct development of the gonadal medulla into testes. And testicular development does not require male sex hormones such as testosterone. So the testes then begin to secrete 3 hormones. The first hormone is the anti-Müllerian hormone, and it is secreted by the Sertoli cells. This causes the embryonic Müllerian ducts to regress. The next hormone are androgens secreted by interstitial cells, and these include testosterone and its derivative dihydrotestosterone. So testosterone converts Wolfram ducts into male accessory structures such as the epididymis, vas deferens, and the seminal vesicle. And it is also responsible for the migration of the testes from the abdomen into the scrotum. And the dihydrotestosterone is responsible for differentiating the external genitalia. So female embryonic development, so there is no SRY gene. The bipotential gonads develop into ovarian tissue with the absence of testicular AMH. The Mullerian ducts develop into the upper portion of the vagina. They develop into the uterus and into the fallopian tubes. With the absence of testosterone, the Wolfram ducts degenerate. And then the absence of DHT, the external genitalia take on more female characteristics. And this will complete my week 13 report.